All right, now I'd like to welcome up Manesh Girn, who is the Chief Research Officer for EntheoTech, and he will be presenting on ketamine antidepressant mechanisms.
Great, thank you so much for the introduction, Sonia. So my presentation is going to be on the mechanisms of ketamine. Anita did mention them briefly, but I'm going to be diving into a lot more detail. So disclaimer, this is aimed at a medical audience, so if you don't have too much of a background in neuroscience, it might be a bit technical. But hopefully it still retains your attention to some degree. Okay, so just because everyone's doing About Me slides, I thought I would include that for myself too. So I'm Chief Research Officer at EntheoTech. In the last year of my PhD in neuroscience over at McGill University in Montreal. I've been on a bunch of peer-reviewed publications, including on psychedelics, as well as stuff on mind wandering and daydreaming, and also the default mode network, for anybody who knows what that is. And I'm currently collaborating with some leaders in the field, looking at basically the brain mechanisms of psychedelic drugs, mostly psilocybin and the other classic psychedelics, but also ketamine is also a big interest of mine. Just a quick overview. So I'm going to be talking about a theory of depression that focuses on neuroplasticity instead of neurotransmitters per se, and then kind of bridging that into the neuropharmacology of ketamine, and then looking at ketamine's antidepressant mechanisms, drawing on the first two sections. Okay, so before I dive into the neurobiological mechanisms, I want to highlight the important point that they're not the whole story with ketamine. So like how it's typically construed, and this has been mentioned, is that ketamine is this purely pharmacological treatment that affects your brain, and you might have these dissociative side effects. And I think that's kind of a untenable view these days empirically and also theoretically with the rise of psilocybin-assisted psychotherapy, which really emphasizes these, what you would call extra pharmacological factors, contextual factors, the alliance with the therapist, and require psychotherapeutic support and a focus on the actual experience. And there's evidence suggesting that ketamine is best viewed through this lens as well. So there is evidence. I'm going to walk you guys through this here. So psilocybin is in red and ketamine is in green. First, what this is a 94-item questionnaire comprehensively mapping out altered states of consciousness. It's the Altered States of Consciousness questionnaire. And it's used with different drugs and also non-drug states as well. And it has 11 dimensions. It's a very complex scale that's been psychometrically validated, etc. So if you look in ketamine, which is green, disembodiment, which you could roughly understand as dissociation, is very high. Yeah, it makes sense. But there's also a whole bunch of other things that are above, let's say, 25% of the maximum, which also capture it. For example, the spiritual effects, experiences of unity, pretty high. Spiritual experience, pretty similar to psilocybin. And then insightfulness, pretty similar as well. And so you can see this complex phenomenology of the ketamine experience, which is actually pretty similar to psilocybin. And by reducing it just to dissociation, we're not really doing it full justice. And not only that, empirical evidence does suggest that mystical type effects with ketamine correlate with depression reductions, and there's also evidence they correlate with cocaine abstinence in cocaine addicts as well. So this is all just to say that psychological or experiential or experience-based mechanisms are also important for lasting outcomes. And this is why at EntheoTech we use adjunctive interventions and psychotherapeutic support as like an integral part of the treatment. First, I want to position ketamine in relation to SSRIs and standard antidepressant medications, right? So these medications were developed under the context of the so-called monoamine theory of depression, which originally was like the serotonin imbalance theory of depression. It's the same kind of thing. So how did we find antidepressants in the first place? And actually, it was very incidental. So these two very early ones, imipramine and ipronizid, very hard to pronounce drugs, but were discovered in the 1950s, and they weren't designed to reduce depression. It was incidentally found by physicians that they reduced depression. For example, ipronizid is a tuberculosis medication. It's a completely different indication, but they found that it was reducing depression symptoms. And then subsequently, they were like, okay, they're doing this, they're reducing symptoms, how are they working? And they found they worked on the serotonin system. So then, after the fact, they said, oh, then perhaps serotonin deficiencies are found in depression, and this is normalizing some pathophysiology, right? But that's not really a tenable inference. It's like saying somebody has a Tylenol deficiency because Tylenol cured their headache. You know, it's a similar thing there. And so a core claim of this whole theory is that that depression, as I said, is a result of deficiencies in serotonin, and then later they added some other neurotransmitters into the mix too, hence monoamine theory because they're all monoamine neurotransmitters. And again, it's assuming that there's some pathophysiology, there's some pathology in your brain where you have reductions in these neurotransmitters, and these drugs are fixing that existing pathophysiology. However, I mean, there's no reliable evidence that people who have depression consistently have lower amounts of these neurotransmitters. And there's also no reliable evidence that these neurotransmitters, that these SSRIs and drugs help people by increasing neurotransmitter levels. And part of the evidence for that is that neurotransmitter levels increase pretty highly acutely, but you don't get benefits for 4 to 6 weeks in most patients. So clearly it's not just the amount of these chemicals that are related to efficacy. And so this whole claim about it normalizing pathophysiology is not really true. And not only that, 30 to 40% of patients don't even respond to these drugs. So there's a lot of people not receiving the treatment they need pharmacologically. And also, it goes without saying, you have to take these kinds of drugs every day. And they have a pretty bad side effect profile with weight gain, sexual dysfunction, and this kind of emotional numbing. And it's, although this was kind of not acknowledged for a while, it is a case that if you're chronically using these for a long period of time, withdrawal is pretty harsh. So it's hard to stay on. You need to stay on, it's hard to come off. And so this is all to say that the monoamine theory and SSRIs and tricyclics and these drugs are not really that mechanistically well-founded. And also the existing drugs leave a lot to be desired. And so then what's like an alternative view of depression that is more mechanistically well-founded, more in alignment with empirical research? It comes from a lot of research suggesting, first and foremost, the reciprocal relationship between depression and chronic stress. You could see somebody who's under a lot of chronic stress might develop depressive symptomology over time, and if you're depressed, you're going to be chronically stressed as well. And so there's this reciprocal relationship. And there's a wealth of evidence from animal studies suggesting, here's where it gets more technical, wealth of evidence from animal studies that chronic stress leads to synaptic atrophy or neuronal atrophy. It just makes neurons a bit more withered up and impoverished. And particularly, this happens in the prefrontal cortex, a place in the brain involved in these kind of higher order mental functions, your attention and coordinated behavior and ability to plan and do multi-step like sequences of action, and also the hippocampus, which is involved in memory, as well as other things. So these areas involved in these fundamental processes are becoming atrophied with chronic stress. And we know this from a lot of research. And specifically what happens is on the neurons, what's targeted is the dendrites. So a dendrite is the receiving part of the neuron that receives impulses from other neurons. And basically, the dendritic length, number, shape, and size of spines is reduced, and is a reduction of expression of glutamate receptors, which allow for transmission to occur. And this results in reduced synaptic strength. So basically, these neurons are less able to do what they're supposed to be able to do, which is receive electrical impulses from other neurons. And there's evidence that once this dysfunction happens, particularly in PFC, mice, they show depression-like behaviors. And also, moving into humans, there is evidence that patients with major depressive disorder have reduced brain volume in the prefrontal cortex and hippocampus, so these two regions that are implicated in animal research. And the thought is a similar thing is happening as humans, and we can only detect it at this higher kind of, like at the level of regions and not actual neurons and cells. But it does seem to correspond as well. And so, basically, what this leads to is a theory of depression where rather than focusing on neurotransmitters, instead it's looking at the neurotoxic effects of sustained chronic stress, which affects the brain in terms of glucocorticoids, such as cortisol, which result in neurotoxicity via increased glutamate and excitotoxic processes in the brain. And particularly the PFC and hippocampus are very vulnerable to this. And so this leads to this neuronal atrophy where neurons are no longer really able to do what they're supposed to be doing. And it impairs neuroplastic processes contributing to changes in connections between these neurons. So think of like a rigid way of thinking or seeing the world is being exacerbated or facilitated by this neuronal atrophy. And furthermore, it creates this circuit dysfunction in circuits which regulate our thinking and interaction with emotion and regulation of our emotion, so-called cortical limbic circuits, become dysfunctional because of this atrophy, again, due to chronic stress. And so the idea behind this theory is that rather than boosting our serotonin levels, or whatever the monoamine theory would tell us, that this tells us instead that drugs which help restore PFC circuit function by boosting neuroplasticity and kind of boosting these neurons and these dendrites will reduce symptoms. And this is where these novel fast-acting glutamatergic drugs, which work on the glutamate system, which, as I'll say, is very intertwined with neuroplasticity, come in, and obviously ketamine is one of these. And so with that understanding of depression in mind, what are ketamine's antidepressant mechanisms on the level of the brain? And so first, Anita kind of covered some of this stuff. Ketamine is a very complex drug in the way it affects the brain. It affects all sorts of receptors: glutamate, opioids, sigma, serotonin, dopamine, and so on. But there is evidence suggesting that glutamate might be the most important for antidepressant effects. And this is to show you, in red is glutamate, so neurons that use glutamate to activate. And you can see in the prefrontal cortex, there's a lot of projections going down to subcortical or maybe more primitive regions involved in emotion in the way that it regulates them and keeps them in check in some extent. So glutamate is very important for this ability for the prefrontal cortex to regulate the rest of the brain. And what ketamine is an antagonist at a particular glutamate receptor subtype called the NMDA receptor. And let's talk about that a little bit more. So glutamate is the main way that the brain communicates between neurons. It's excitatory. It makes neurons activate. And the two main receptor types important for the present context are AMPA, which is the main receptor type that glutamate uses. And the thing with AMPA is it comes in and out of the synapse in a very fast way where it can regulate how strong that synapse is. So if there's a lot of AMPA receptors in the synapse, it's going to be more sensitive to being firing. That connection is stronger. But if there's less, then it's weakened. And they're very easily be able to cycle in and out of the synapse. So that's AMPA. And then NMDA is kind of a complement to AMPA, and actually can only become activated when the neuron is already activated. But also, importantly, when NMDA, it's located in the actual synapse, but also extrasynaptically on kind of like the cell body. And it does different things depending on where it is. So those are the two main receptors that are important. And so, okay, so then what does ketamine do exactly? So at sub-anesthetic doses, and that's important because there's a lot of dose-dependent effects, ketamine preferentially seems to bind to NMDA receptors located on GABA interneurons, because there's particular subunits located in that area that have higher affinity for ketamine. So basically it's blocking a neuron that is inhibiting a glutamate neuron, you could say. And so by blocking that, it disinhibits that neuron and allows it to release more glutamate. But the interesting thing is, when this happens, there's like a big surge of glutamate that comes in, and this does a variety of things. So one of the things it does is it activates AMPA receptors, and these AMPA receptors activate neuroplastic processes, such as releasing brain-derived neurotrophic factor, which then go up and then affect that same receptor and lead to neuroplastic processes that bring more AMPA receptors up. So it's like this reciprocal thing where glutamate is activating this neuron, and by activating this neuron, it boosts neuroplasticity and makes that neuron more sensitive to the glutamate. So you get this huge boost of glutamate-induced neuroplasticity, again, in the PFC. And moreover, there's a whole other mechanism too with receptors located outside the synapse, extrasynaptic. By inhibiting those, there's another, independent of that glutamate release actually, another way that ketamine can boost neuroplasticity. And as I showed you before, why is this relevant, why is it useful, is that increasing these neuroplasticity-related processes through these two different mechanisms, ketamine reverses the effects of chronic stress that I was talking about before. In particular, there's a lot of evidence showing that ketamine increases the number and function of dendritic spines. So if you remember, those are the things that chronic stress is really targeting. And again, these are the receiving parts of the neuron that allow it to be influenced by other neurons and to propagate signals. And furthermore, what this figure is showing is like this is, you could say, after ketamine, this is before ketamine. There's more AMPA receptors going in here, and it's actually widening the synapse, so again, making it more sensitive to being activated. Perhaps more like it should be in health, as opposed to being atrophied. And we know furthermore that if you block through if you genetically or pharmacologically block aspects that are related to neuroplasticity, you block the antidepressant effects. So there's a pretty strong relationship between these neuroplastic processes induced by glutamate and the antidepressant effects of ketamine. And so that was with animals. And then also there's evidence in humans, for example, that in people who go into remission after treatment, have their hippocampus increases in volume more. So if you remember, the hippocampus is one of the stress vulnerable areas. And also, area in the inferior frontal gyrus, which is, you can think of it as the prefrontal cortex, also increases in size in a way that correlates with depression reductions. So this is kind of human-based evidence showing the translational likelihood of these animal findings. And so this is kind of my summary slide now. We could say that ketamine is an exemplar fast-acting glutamatergic antidepressant that catalyzes neuroplastic changes in a manner that seems to reverse the deleterious effects of chronic stress. And this is kind of the way this happens, we would think. There's these cellular molecular effects happening at the neuron level, at the level of dendrites, like I said. And this might translate into macroscale structural change, which is detectable in humans. And this might help normalize circuits of interconnected brain regions which were previously unable to function in the way they should be. And so we can say that ketamine seems to be mechanistically more well-founded based on preclinical and human studies than the monoamine antidepressants, and very much aligns with nascent neuroplasticity/glutamate perspectives on depression, which are really replacing the monoamine theory just on the basis of empirical evidence these days. And again, so the treatment that seems to actually counter genuine pre-existing depression-related pathophysiology. And yeah, that's it. Thank you.